All right, it's my pleasure to introduce Yesutor Soku. He is a PhD candidate at the College of Veterinary Medicine at Tuskegee University under the direction of Dr. Muhammad and in collaboration with Dr. Miles Lang. His research interests include aquatic animal health and food fish safety. He is researching the pathogenesis of motile Aeromonas septicemia by investigating the role of dietary status in the disease process at the tissue and molecular levels. Looking forward to your talk.
Thank you, Dr. Nick. Good afternoon, everybody. I am presenting on the temporal dynamics of motile Aeromonas septicemia in channel catfish with varied skin injury and feeding conditions. We've had several talks on Aeromonas. So by introduction, I'm just going to summarize the intro. We know that motile Aeromonas septicemia is mainly caused by Aeromonas hydrophila, but other Aeromonas species, including caviar and Veronae biovasobrae, are implicated in MAS infections. We know that the first reports of the virulent strain of Aeromonas hydrophila, which causes MAS, started in 2004 on a catfish farm in Mississippi, but major outbreak started in 2009, and up till now, it's still been ongoing. As a result, we know that there is a 2% reduction. So from 2022 to 2024, there has been a 2% reduction according to NAS. And it's not just attributed to Aeromonas, but a whole lot of challenges within the industry, including Aeromonas hydrophila. So my research concept was to obviously investigate literature, and we found out that some portals of entry has been explored, and by my colleagues in USDA ARS, they explored various portals, and then they found out that the most probable route of infection was during a fin clipping where the skin was exposed to the bacteria. My same colleague Jean and colleagues explored the number of VAH bacterial cells in the various tissues, and then they found out that they were present in several tissues, including the intestines and then the kidneys. Western colleagues down in Mississippi also explored the histopathological lesions in failed isolates, and then they saw that unlike previous motile Aeromonas septicemia infections, the VAH specific MAS diseases had very specific lesions within the GIT. So we hypothesized that obviously the skin is playing a role in the pathogenesis as well as the feed, because farmers usually complained of high mortalities, especially after they fed the fish with feed. So in that regard, we decided to set out with various objectives, including to determine the survival rate of these MAS infected fish, and then to look at the histopathological lesions. But this time around, we were going to score them so that we understand whatever was happening at the tissue level. So with the methodology, we're using catfish, and then we established the numbers of catfish that we were going to use based on pilot studies and various statistical analysis. The average weight of the fish was 35 grams, and then we kept the fish at water temperature of 28 degrees Celsius. For the experiment, we kept a flow rate of 0.5 liters per minute. The fish was actually fed on commercial diet at 3% body weight, and we kept a photo period of 12-hour light to 12-hour darkness schedule. Before we started the experimentation, we randomly sampled 10 fish to check whether they were absent for virulent strain of Aeromonas, which we actually did, and then we found out that none of them had the VAH strain that we use. So the VAH strain that we used is the ALG 15097, which was found in Alabama. So what we did for the objective one, which is the survival analysis, was we cultured them and incubated them. We determined the concentration, which was 1.5 times 10 to the 7th CFU per ml. And then we performed the challenge in the flow through tanks that we see in our picture. We had 4 treatment groups because, like I said earlier, we were exploring how the skin was a determinant as well as how the feed was also a determinant. So the first group was fin clipped and fed. The second group was not fin clipped, but they were fed. The third group were fin clipped, not fed. And then the last group were not fin clipped and they were also not fed. So we observed them over a period of 5 days, but we actually ended up halting the experiment because by 72 hours, we had almost all the fish dying with this particular strain. For the second objective, which was the histopathology study, a similar approach was used. But after the challenge with the 4 treatments, we sampled the stomach and then the intestines, fixed them in neutral buffer formalin, and then embedded them in paraffin wax, cut them in sections, and then we observed the histologist, the slides on the microscope and recorded the data. So with our results, we did a Kaplan-Meier survival study for the various observations that we saw. And then we found out that the fin clip groups actually had the lowest survival. And then the statistical difference between the fin clip groups were not that much, which supported the already explored literature that fin clipping actually was the major route of or the major determinant in the pathogenesis of this particular disease, because we realized that those that were not fin clipped actually had a higher level of survival. How about feed? We realized that feed was just playing a role in modulating the disease, and I'm going to talk about that more in the histopathology section. We also did this was a risk ratio using the no clip, no fed as the reference, because out there in the field, the bacteria will be exposed to the fish will be exposed to the bacteria. So we realized that again, the fin clipped groups actually had the highest risk of mortality compared to those that were not fin clipped. When we did the histopathology, now this was very amazing. So we saw various lesions in both the stomach and the intestines. I'm going to some portions of the intestines over here. So this was 2 hours post-infection. And then for the various treatment groups, A was the fin clip fed, the B was the fin clip not fed, the C was no fin clip but fed, and then the last one was they didn't have any fin clipping or feeding at all. We realized that it was just mild to moderate edema that were present with minimal lymphocyte infiltrations. However, at 4 hours, it began to increase where we were seeing more edema. And then finally, at 8 hours, we saw that the edema was actually very, very pronounced. And then there were degenerative changes even in the superficial epithelium. And there was also hemorrhage that was present within the epithelium. Now, this is actually showing the remarkable lesions within the GI tract, but we decided to further score them. And with the scoring, we developed a semi-quantitative grading system by the help of a histopathologist. And then we found out that the group that was actually fed had more lesions within the gastrointestinal tract compared to those that were not fed. And that is why the lesions were more pronounced in these groups than those that were not fed, which signifies the role that the feed was playing in modulating the disease. Now, why do we think that this is happening? It is because based on normal physiology, we understand that when you feed, blood rushes to the gut. So we understand that the Aeromonas hydrophila is bloodborne, it's in the blood. So with more blood rushing towards the gut, we're going to be seeing a lot more of the bacteria causing its damage to the gastrointestinal tract system. So comparing the various groups, the fed treatments, we are trying to now understand the pathology. So imagine that the fish is out there in the field and exposed to this bacteria. What is happening is 4 hours post-infection, there are significant changes that are happening in the tissue level, including edema, hemorrhage, infiltration of inflammatory cells, and necrosis. And then at 8 hours post-infection, we begin to see mortality. However, if the fishes are unfed and they are still exposed, we see that because there are no significant mortalities at 4 hours and 8 hours, the mortalities become delayed. So the mortalities happen a little after 8 hours. So it's mainly at 12 hours and more significantly at 24 hours. The future works now is, since we understand what is happening at the tissue level, we want to explore what is happening at the molecular level by doing a transcriptomic study. So we've taken samples and we're going to run RNA transcriptomic studies on this to understand the cells that are regulated both down and up within these samples that we have taken. So I have a lot of people to thank and agencies that funded. So this is my work and my references, and thank you for your audience.
Excellent presentation. That was very well done and an interesting project.
Thank you.
Yeah, folks, if you have questions out there, feel free to unmute yourself or you can drop them into the chat. Okay, we got one here in the chat. Yesutor, do you believe that the gut pathology is due to a toxin?
It could be, but looking at the I believe the toxins would have caused more changes, like the edema will probably be marked, and also depending on the specific toxins. Based on what we know so far about the virulent strains, I don't really see the lesions that we are seeing to be correlated with toxins, unless the toxin is not like very, doesn't cause very adverse effects, but that is actually a possibility though.
Mm-hmm. All right, real good. Thanks again.